All right, let's go ahead and start lecture. First of all, I have lied. The unit study guide is not yet posted, but I'm keeping this up here to keep myself accountable. I should have that posted by the end of the day. But if you notice at the end of all the slideshows, there's been a slide that says study guide for these slides. Basically, I'm just comparing all those slides to make that unit study guide. Let's see. I think that's all I have for announcements today. I'm hoping to get through Meiosis today. If we don't, that's okay. You might have noticed on the schedule we are a little bit behind. That's fine. I'm not going to try to rush us through the content because we still have after Thanksgiving. We have a little bit more wiggle room. You will notice that on the schedule I have moved, and I put this on the homework side, I have moved content check number 7. Right? Originally that was due next Tuesday. I've decided to move it to the next unit, all right? Because I think we have more time in the next unit and it just makes more sense there. So if you have that down in your schedule, you can erase it. There's a new due date up on the schedule that's posted on Moodle, but it's after this next exam. All right, we do have a content check today that we'll do in class. So if I look like I'm running out of time, like raise your hands so I make sure that we get to this content check. But I've been doing a pretty good job of keeping an eye on the time. So, all right, Tuesday we talked about mitosis. Right? Today we're going to talk about meiosis. And so that's where we will start class. All right, so mitosis, if a cells are going to reproduce using mitosis, that would be asexual reproduction. Okay. And so during asexual reproduction, we get offspring that are genetically identical to the parents, or to the reproducing individual, I should say. Right, so they're genetically identical, which works for some species. Right, some species it's okay to asexually reproduce most of the time. But for many other species, many other organisms, we need to go through sexual reproduction. Simply because the offspring are going to have unique combinations of genes, right? We're going to increase our genetic diversity, right? So genetic diversity is very important. There's an example of the banana plant, right? Most of the banana plants nowadays are clones, so they are genetically identical. And now there's a disease that are affecting banana plants. And because they're all genetically identical, they don't have any genetic diversity for some of them to resist the disease. They are all susceptible. Right? So genetic diversity is important not only for fighting disease, but just in giving us some diversity of features, whatever that might be. All right.
Local food. Yay!
Yes. So sexual reproduction, genetically identical individuals. Right, sexual reproduction, unique combinations of genes. They are unique individuals in terms of their genetic makeup. All right, now there are some advantages and disadvantages of sexual reproduction, right? In fact, there's quite a lot of disadvantages, right? It can be complicated. It can be costly for these organisms, right? You have to invest time into finding a mate, right? Energy into finding a mate. That's energy that you could be using hunting for food, right? Or feeding yourself here. You have to invest into mating strategies, right? So I don't know if you've ever watched videos of like the birds of paradise where they have these very elaborate mating strategies. These birds will take time to find like certain colors of objects and build elaborate displays in an area or clear out a clearing so that they can be seen more easily. That takes time, that takes energy, all right? Again, time and energy that could be spent elsewhere. I'm actually going to hold off on questions for now. They can be potentially dangerous, all right? So when you are going through sexual reproduction, you might be more visible to predators, more vulnerable, right? That's potentially dangerous. You could get your disease from the mate, right? It's also very dangerous. And finally, it's not always successful, right? Sometimes you invest all this energy and time into finding a mate and going through your mating strategies, and it doesn't end up in any offspring, right? So there's a lot of disadvantages to sexual reproduction. But the one advantage that it provides is that it increases that genetic variation. And that one advantage is huge, all right? We need genetic variation.
I would say another advantage too is that usually animals that do sexually reproduce usually is that a lot of them rely somewhat on another parent to be around to raise young, because the young are more likely to be born altruistic.
Yeah, that is very true. So I did not add that out here, but there are some like social advantages, right, to sexually reproducing in the animal kingdom, that you might have two parents to help raise the young. Yeah. All right, so lots of disadvantages. It's costly, it takes time, it takes energy, right? It's potentially dangerous, but the payoff is great, right? We're increasing genetic diversity, and that is key. All right, so we're talking about sexually reproducing organisms. I have to briefly mention that not all organisms are going to do this the same way and have the same type of life cycles. Now, if you go on to take organismal biology next semester, you'll dive into these life cycles in way more depth. But I just wanted to briefly introduce them, right? Some organisms can be a diploid dominant species. Right, so remember, diploid is two chromosomes representing a set. They can be haploid dominant species, so one chromosome representing a set. Or they could alternate between the two. And these are the most common life cycles. Right? I realize some of you are taking notes. But my next slide is going to demonstrate these terms a lot more. Right, so if we're talking about life cycles, it's the series of events that produces another generation of individuals. All right, so what do I mean when I say they're diploid dominant species or haploid dominant species? Right? I mean that's the diploid level that they spend most of their time in, that their cells are most likely going to be. Right, so most animal species, we are a diploid dominant cell, or diploid dominant organism. Right, our cells, if you were to take a sample of cells from our body, they are almost always going to be diploid. Right, we spend most of our life cycle as a diploid dominant species. It's only when we go through the process of meiosis to produce gametes, right, that our cells are haploid. Right, so it's just a sliver of time in our life cycles, in other mammals' life cycles, where they have haploid cells. Right? Most of the time their cells are diploid, so they are a diploid dominant species. There are other species that are haploid dominant. A lot of fungal species tend to have haploid dominant life cycles, where again, if you were to take a sample of cells from a fungi culture, more than likely you're going to find haploid cells. That is the stage, the ploidy level, that these cells spend most of their time in. Right? And again, it's not until they reproduce that they briefly enter into this area where these cells can be diploid. Right? They have a very unique type of reproductive cycle. And then finally, many plants kind of alternate roughly the same between being haploid or diploid. We call this alternation of generations, right? They spend a considerable of time being a diploid dominant species, and then they can spend a considerable amount of time being a haploid dominant species. They alternate between the two, right? So all of these life cycles, they vary based on the species that we are looking at. Now again, we're focusing mostly on mammals here, so we're going to look at these diploid dominant life cycles and focus on organisms that most of the time our cells are going to be diploid. Right. My next question where I have a stop and pause, I think I'm going to lower the shades a little bit because the sunlight is starting to come out. All right, so if we're going through sexual reproduction, we are going to go through meiosis. Right? So meiosis is the process of creating those genetically unique individuals, right? But we're going from a diploid cell to a haploid cell. We are halving the amount of genetic material in the daughter cells. Right, so we're creating haploid cells. We're actually going to create 4 daughter cells, and we'll get into that in a minute here. Right, these haploid cells are then important in creating the new individual. Right, so once these gametes have been made, right, again in mammals that's typically egg and sperm, those 2 haploid gametes will unite. And when 2 haploid gametes unite, they create a cell called a zygote. Right, a diploid cell called a zygote. It's basically the first cell in this organism's life. Now, maybe on your screens, because the sunlight's kind of washing this out a little bit, I have a life cycle of like human development here. Right? And you'll notice that the very first cell I have labeled as the zygote. Right? So humans, we have 23 sets of chromosomes, but 46 total chromosomes, because we're a diploid species. So each gamete has half the amount of the chromosomes of the original. So each gamete would have 23 chromosomes. And when each gamete unites to create the zygote, then we become a diploid cell, and we have 46 chromosomes. Right? Now, once the zygote has been created, then this zygote can go through the process of mitosis and develop into the next individual. All right, so I want to do a group review here, and I'm going to lower the blinds so we can see the screen a little bit better here. But I'll give you a couple minutes to ponder these questions. Maybe they're relatively easy. All right. Let's talk about the advantages of, let's do sexual reproduction first, because we just went over that. What is the advantage of sexual reproduction? And it's a big one. Genetic diversity, yes. Also having some social advantages of having, a lot of times, 2 parents around, not always in the animal kingdom. But what would be the advantages of asexual reproduction? Yeah, less energy can be used. Why is less energy used?
You don't have to put any effort in finding a mate.
Exactly. Yeah, you don't have to put in any effort to finding a mate. What other advantages might there be? Yeah?
Usually in asexual reproduction, a bigger feature is that usually organisms are born fully grown.
Yeah. A lot of times organisms can be born fully grown, right? Born is a term we're using loosely. Yeah.
Yeah, it's a lot, but even that has some disadvantages too, because having one that's born altruistic allows it to become a more complicated organism in the future.
Yeah.
It's more safe.
Yeah, it can be more safe. It's not as dangerous, right? You're not going to be vulnerable. You might not have the opportunity to catch any potential diseases. Yeah. All right. So let's dive into meiosis. Meiosis here has actually two parts to it. So you'll see that I have listed meiosis 1, and then I'll talk about meiosis 2, right? But like mitosis, all right, we're thinking about the cell cycle. This is happening after interphase, right? So our DNA has replicated, our cell has grown, it is ready to divide, right? Once we enter into our M phase here, right, which is going to be meiosis, we're going to start with meiosis 1, right? And this is where a diploid parent cell is going to produce 2 haploid daughter cells, right? Homologous chromosomes are going to separate, right? So notice in mitosis, I emphasize that it was sister chromatids that separated. Here it is homologous chromosomes that separate first. And we're going to exchange genetic material to create some genetic diversity. Right, so again, this is just a very broad overview. We're going to go into much more detail here. Now at the end of meiosis 1, cells are immediately going to go into meiosis 2. They're not going to go back into interphase. They're just going to go right on into meiosis 2. Right, so at the end of meiosis 1, we've gone from a parent cell to 2 daughter cells that are haploid. Now in meiosis 2, both of these daughter cells that are haploid, the end result of meiosis 1, they're going to go through each separately this stage of meiosis 2. Right, in meiosis 2, we're going to get sister chromatid splitting, right? So now it's very similar to mitosis. And we're going to produce 4 haploid daughter cells. Right? And we can see that on the image here, right? The result is 4 haploid daughter cells. All right, so I like to compare and contrast meiosis 1 with meiosis 2 compared to mitosis. And I think this slide gives you a good, broad overview of what the differences are. It tells you when homologous chromosomes separate, when does a cell go from diploid to haploid, is it meiosis 1 or meiosis 2, and when our sister chromatids separate. All right, so let's dive into meiosis 1. Meiosis 1 is I shouldn't say very different, but it's more different from mitosis than meiosis 2 is, right? But it still has the same phases. We still have prophase, prometaphase, metaphase, anaphase, telophase, right? We still have those stages. They still go in that order, but there are some extra things that occur, some different steps within those phases. Right, and again, I'm going to draw this out as I speak. That way you have a consistent visual over here. All right, so prophase 1 we would call it because we're in meiosis 1. Right, again, that nuclear envelope is going to break down. Very similar to mitosis. All right, so I'm going to draw it as a dashed line here. All right. Now what's different in prophase 1, actually, let me back out. Let me stick with what's similar first. All right, so nuclear envelope starts to disintegrate, chromosomes start to condense, and the mitotic spindle starts to form. Right, the same 3 things that happen in mitosis. So let me draw in my mitotic spindle starting to form. Let's do yellow and blue, very bright and dark contrast here. And I'm just going to draw, I'm going to draw 2 homologous pairs here. And again, I'm going to go with the big and short, so size is indicating homology. All right, my chromosomes are condensed. Now what's different in meiosis 1, prophase 1, is that these chromosomes that are homologous are going to pair up. They're going to form what's called bivalents or tetrads. And I often use those two words interchangeably. They mean the same thing, right? So I draw my chromosomes here. They're going to pair up. And I should really draw them a little closer because they are pairing up. In these tetrads, they're really close to each other. Right, so notice in mitosis, we didn't have them pairing up. This is very different. Now, when they pair up, right, they form what's called a synapse. Right, so on my image up here, you can see this synapse is when they're really close to each other and they're kind of overlapping almost. Right? And when this synapse occurs, we get something called crossing over. Right? In my last image up here, you can see that after this synapse occurs, we have swapped genetic material. So basically, each chromosome has broken off a chunk of it and swapped it with its other homologous chromosome. Right? So siglets or chromosomes are exchanged with each other in this crossing over. Right, so I'm going to draw this in a different cell here. So I'm going to label this prophase 1. I'm going to say it's early prophase 1. The next one will be late prophase 1. Still have my cell here. My nuclear envelope is still disintegrating. My mitotic spindles are still in the process of being formed. But what's different is that I have crossing over. All right, so I'm going to draw it as we swapped a little bit of our colors around. Remember, colors are representing chromosomes from different parents here.
And chromosomes aren't different and colored in real life?
Not in real life, but it really helps us remember when crossing over occurs. Now, it's going to happen for all the bivalents, right? So I'm going to do this for my small pair as well. All right, so crossing over has occurred. This is huge. This is what creates the genetic diversity, right? This is where we're swapping around some different versions of genes, right? So that we're creating new combinations of genes. Right, so if we look at crossing over, it's also called homologous recombination. Right, that's more of like the proper terminology to it. Right, if we look at crossing over and what exactly is being swapped. Right, here I have a set of homologous chromosomes. Right, on the chromosomes, each of these colors represents a gene. Right? So in this example, we're looking at the gene for earlobes, and that is this kind of like this darker blue color, this third color down from the top here. That is the gene. That is the area of your DNA that encodes for your earlobes. Okay. Now remember, I say a homologous pair. It has the same type of genetic information, but it has variation. The variation that comes from mom and dad, or the different versions of a gene, we would call an allele. Right? So when we're talking about earlobes here, we can have the allele for earlobes to be attached, or for earlobes to be unattached, or free earlobes. Right? You can feel your own earlobe and see if you have attached or unattached earlobes. Right? So we have our gene, and codes for the genes for our earlobes. The alleles is the variation. The alleles is whether or not our earlobes are attached, right? Or whether they kind of loop down, or they're free hanging a little bit here. Right, so different versions of a gene. There's also one more term on this slide that should be new here. Okay. So I say an allele is one of two or more versions, if we're dealing with something that's like triploid or tetraploid, of a DNA sequence at a given loci, right? LOCI. Right? A loci is simply the area. It's the location of these genes. Right? So if I was a scientist, I was trying to describe what gene I'm working on in this chromosome pair, I would determine the loci, right? The specific loci, and that's what I would pass along to my fellow scientists. Right, so crossing over, we're swapping around these different alleles, right? So you might have a chromosome that's mostly from mom, but during crossing over, right, we've swapped for an allele that came from dad. Right, now because this is such a big process, I have another screen here that helps us kind of visualize this. In fact, I have a few different slides that help us visualize this here. Right? So, we're crossing over. Homologous recombination. Right? This is the exchange of genetic material, and it's between non-sister chromatids. Right? So remember, a sister chromatid is identical. Right? Identical to either side on that X. If crossing over were to occur with those sister chromatids, it would not yield any genetic diversity because those sister chromatids are already identical. So crossing over is going to occur between homologous chromosomes. When crossing over occurs, we say the result are recombinant chromosomes. One second. And again, I have an image here, right? I have two homologous chromosomes. I can see they've indicated where the swapping over is going to take place. They've kind of had some dashed lines. And when crossing over is done and complete, we get our recombinant chromosomes, right? So an allele that might have been mostly, I should say a chromosome that is mostly from mom, is now going to have some alleles from dad, right? We're creating that genetic diversity. Lucy, we had a question?
Yeah, I'm curious about about loci, right? And
Yeah,
I'm
loci.
I'm curious. Do all DNA sequences within a loci affect the same location?
Affect the same location. I would say all DNA in a chromosome has a specific loci, and I think it's a numbering system that is used. It's been a while since I've looked up a loci location on a part of a chromosome. So all chromosomes have loci, and loci is kind of like, if you were to think about trying to use GPS, we're using coordinates. It's kind of like the coordinates for a chromosome.
See, I was more. In other words, not all loci will. In the example that we were given, not all of the loci within that sequence, within that gene,
Would be, repeat the last couple words.
so not all. Not all of the loci within the homologous chromosome. So that whole sequence does not. Only part of it affects the ear. The whole thing doesn't.
Right. So the entire chromosome is going to have many different alleles, right? And so each allele will have like a given loci. Like this location is towards the upper part of our chromosome. Maybe the alleles down here might be for height, right? So each chromosome has many alleles. Yeah, and that's a good point to emphasize. All right. Crossing over. There's often misconceptions about crossing over. I want to tell you what it does not do, right? It increases genetic variation, yes, but it does not create new genes, right? So if we had to create a gene for, maybe all of a sudden humans develop the ability to have different colored fingernails, right? We are not creating a new gene to have different colored fingernails, like blue, purple, and yellow, right? We're not creating new genes out of the blue. And what it does not do is reduce or increase the likelihood of a mutation, right? Swapping over does not determine if a mutation is going to arise. If we get mutations, that's typically going to be in DNA replication, or when we talk about next week, transcription and translation, right? Now there are some abnormality things that can happen. We might get to that at the end of our lecture slide today, we might not, right? What it does do is create new combinations of your parents' genes within your gametes. Right, so again, genetic diversity. All right, so crossing over, remember we're still in that first phase of meiosis. We're still in prophase here. I wanted to go over this a little bit more, yes?
Just to clarify, the crossing over does not happen in meiosis.
Correct. Yep. Crossing over is unique to meiosis. All right, so we're going to look at two homologous chromosomes and follow these steps of crossing over to kind of give you a little bit better of a visual here. Okay. We have our tetrad, or our bivalent. Okay. We have our synapse, also called a chiasma. Right? There's always like several terms that mean the same thing in biology. Makes it confusing. We gotta flow with it here. But it creates this synapse, this chiasma, where there's a little bit of like connection between the two homologous pairs. Now at this chiasma, if we were to look at this drawn out with our alleles, right? So we have a C allele and we have an E allele. Right? We're showing the differences in the alleles, the variation, by having a capital C and a capital E versus a lowercase c and a lowercase e. Right? So when we have our tetrad in our synapse, creating that chiasma there, it's actually going to physically break parts of our chromosomes. Okay? When parts of those chromosomes break, right, they're going to connect together and swap across. And so basically they flip-flop in place. And so what's left are these chromosomes that once only had, say, all capital letters, right, that version, that capital version of a gene, now has an instance where it has the lowercase version of a gene. And we're creating that diversity. And now we're going to get into more on how this actually gets passed down, like the genetics, Punnett squares in our next unit here. So I don't want to elaborate too much for now. I just want you to know that we're swapping over, we're exchanging genetic information. Right? So ultimately, when these chromosomes finish going through meiosis, right, and at the end of meiosis 2, we get 4 daughter cells that are haploid. Right? We will be splitting apart our chromosomes, homologous chromosomes, and our sister chromatids, and so that each cell will get one part of the chromosomes. Right? So each cell will be different from each other. There's no one cell that will have the same, exactly the same genetic makeup. All right. As we go through this, maybe this will start to click a little bit more. I'm trying to foreshadow a little bit here. So again, we are still in prophase. I want us to do a quick review before we move on to any other stages of meiosis. Prophase is the most common. No. Getting up this morning was a challenge.
See, I didn't my brain forgot when I started.
Okay.
So I got here half an hour early and was like, why is no one here? I was like, should we have an hour? Oh my god, I'm like, oh, really lost it. I forgot someone's here.
Oh, you were up and at 'em. Your brain told you that it was time to go. Maybe it's still like the daylight savings effects.
I was, yeah, last night I got off work at 10, and I went to my clock in my car, and it's like 10:35, and I was like, I did not sit there and talk for an hour and a half.
At least it's the fall one, right? Instead of fire.
Oh, you gotta go the hour.
I think that one has to be a
Oh,
more.
oh, yeah, that. So is also.
What, really?
I mean, before one day you start.
All right, so these should be relatively easy because we just talked about it. Right? In what stage of meiosis does crossing over occur?
Yeah.
Prophase. It's the only phase we talked about so far, right? Prophase. Prophase 1, to be specific. All right, does recombination happen between sister chromatids or homologous pairs?
Homologous pairs.
Homologous pairs, yes. We need to increase diversity. If that happens between sister chromatids, it would be pointless because the sister chromatids are identical. And what is the purpose of recombination of meiosis? Genetic diversity, yes. Like I said, these might have been easy, but I felt like we needed a stopping point in the lecture because prophase is kind of complicated. All right, we are now ready to move on to our next phase, right? The next few phases are going to be similar to mitosis, but have a slight variation. So meiosis 1, we're in prometaphase 1, right? Again, the nuclear envelope is completely gone. This spindle apparatus, or the mitotic spindle, is completely formed, and it's attached to our chromosomes. I'm going to draw it out here. It's taking a little bit longer to draw these because now I have to draw crossing over. Keep them in their bivalence. Alright, so I've drawn them next to each other. They look like they're paired up still. They're still in these bivalents. Our mitotic spindle is fully formed. So I have my centromeres here. My microtubules are going to attach to either side of the bivalent. So this is different than mitosis. In mitosis, the mitotic spindle connected, each of them, connected to both chromosomes, right, or all the chromosomes. In this case, we're only connecting to either side of a bivalent. Right, so instead of drawing, say, 4 lines radiating out from the centrosome, I'm only going to draw 2. Right, again, they're attached to these chromosomes at the kinetochore, right, that location where they attach. Right, and pairs of sister chromatids are attached to a single pole. So again, we're attaching to either side of the bivalent. Right? Each half of a bivalent contains sister chromatids. So at this point, these sister chromatids are not identical anymore because crossing over occurred in the previous stage. All right, so everything's mostly the same as mitosis, except for how the mitotic spindle attaches to our chromosomes. All right, so we have prophase where a lot occurred, right? Crossing over happened, that was major. Right, now we're in prometaphase 1, right? Really similar to mitosis, except that these chromosomes, sister chromatids are attached to each pole instead of an individual sister chromatid. Okay, and this image down here, I forgot I had this pop up here. Right, just to compare mitosis, right, sister chromatids are attached to both poles. In prometaphase 1, sister chromatids are only attached, a pair of them are only attached to one. All right, metaphase 1, again, very similar to mitosis, right? We have our cell, we still don't have a nuclear envelope. still have our mitotic spindle. It is still attached to our chromosomes. This more needle here. And just like metaphase and mitosis, we're lining up on the metaphase plate. All right, what's different here is that these chromosomes are lining up in what appears to be this double row of chromosomes, right? Because we're lining up these bivalents. Remember in mitosis, it was just a single row. It was one chromosome, and then another chromosome, and then another chromosome. Here we have two chromosomes in a bivalent lining up against another two chromosomes in a bivalent. So it looks like this double row, right? But we're still lining up in the middle of the cell. We're still lining up on the metaphase plate. Right? In metaphase, this is where we get this terminology called independent assortment. Because the way that they line up on this metaphase plate is completely random. There's not a set order. So for example, the genes are the chromosomes that are mostly from mom are not always going to be all on the right? Or they're not always going to be all on the left. There's no predetermined order. This is all independent. So if we look at this image up on the screen here, we have two possibilities. We have two bivalents. Right now, they did not show crossing over to simplicity. But notice there's one way that we can line them up, where, say, the blue chromosomes, maybe from mom, are all on one side, and the red chromosomes from dad are all on the other side. Or they could have lined up randomly, right? So there's one from mom, one from dad on either side. Completely random. If we take this cell through the rest of mitosis that we'll talk about here, right? After meiosis 1, we'll have daughter cells that look different depending on how they lined up on this metaphase plate. Right? So if they lined up with both colors, or the same colors on each side, right, our resulting cells are going to be cells that have only chromosomes from mom or only chromosomes from dad. If they lined up where there is a color, each color, on each side, right, our resulting cells will have a chromosome from mom and from dad. And if we take this through the rest of meiosis, right, when we get 4 daughter cells, again, we see that the resulting combinations of genes are different once we've gone through all of meiosis. Right, depending, again, all on how they lined up in metaphase 1 on the metaphase plate. So again, this is very random. Right, this is called the principle of independent assortment, because they're assorting themselves on the metaphase plate independently of one another. Right, so when we think about this in terms of humans, right, or diploid species, right, this increases the number of unique variations that we can have in resulting cells tremendously. All right, so if we have metaphase 1, and we say we're a diploid species, so we have 2 to the n power, and n is the number of chromosomes per set, right? Humans have 23 chromosomes in our genome. So that would be 2 to the power of 23. There's over 8 million different combinations that we could have for our cells after they've gone through meiosis, just depending on how they lined up on the metaphase plate. Right? So we're greatly increasing our genetic variation. Now, I don't expect you to know how to calculate this, right? But rather I've shown this slide to illustrate how the power of independent assortment is different. We're really increasing genetic diversity. All right. So we're going to move on. stages of meiosis 1. And again, these happen similarly. So let's start with anaphase 1. Okay. Anaphase 1 is different from mitosis in now that homologous pairs split. Alright, so I don't have my nuclear envelope, I'm not going to draw it. My homologous pairs are splitting apart. Get all straight with our recombination. That's like the trickiest part of drawing out these cycles. Homologous pairs have split, which mean our tetrads have split apart. We no longer have tetrads. Right? They're each going to either side of the cell to either pole in anaphase 1. Again, it's the mitotic spindle coordinating this. The mitotic spindle, as it shortens, it pulls apart the tetrads, right? And it splits our homologous pairs. Okay, so it's not too different from mitosis. Alright, once they have split, and we're ready for our next phase of telophase 1, again, very similar to mitosis. When now the nuclear envelope is reforming, and in each nuclear envelope, right, we have our chromosomes that have split apart. But now, if you look at each nuclei in this phase, in telophase here, right, each nuclei is haploid. We no longer have homologous pairs. Right, we have split them in anaphase 1. We are now getting towards a haploid cell. In telophase, we essentially have one big cell with two different nuclei. Right, now, of course, we have to go through cytokinesis. Right, after cytokinesis, our nuclear envelopes are fully formed. I'm going to draw my chromosomes still condensed, just for simplicity of seeing what is happening here. But remember, our chromosomes have been decondensed at this point. They look like a plate of spaghetti, harder to tell them apart. Right, but the ploidy level of each is haploid, just a singular end. All right. So meiosis 1, we have walked through all the stages. The biggest differences being crossing over occurs in prophase. In metaphase, we have lining up of tetrads, and that's independent. We have independent assortment happening. And then in anaphase, we're splitting apart homologous pairs. And so our end result are two cells that are haploid that have gone through recombination.
I have a question, but it's a little off topic.
Why don't we hold for now?
Yeah, like,
When I'll come back to around you when we do our.
that's essentially what I need.
Yes. All right. Now, meiosis 2 happens very similar to metaphase, or excuse not metaphase, mitosis. You can tell I need a little bit more coffee this morning. All right, so meiosis 2 happens very similar to mitosis. There is no S phase, right? There's no interphase between meiosis 1 and meiosis 2. These resulting cells both simply just go on into the next stage. Okay. The way that they're aligned on the metaphase plate, the way that they are assorted are the same. The biggest difference is that when we get to anaphase, our sister chromatids are going to split. gonna draw this out on the board, but I do need to bring up the screen to do so. So I'm stalling while you all have a chance to write down any notes you want to take. All right, so what enters into meiosis 2 are 2 cells that were the result of meiosis 1. All right, I am going to pull up the screen now. I'll pull it up first so you can still maybe see it. You still need to put the text. Screen is always clean. You have to get it at the right angle, at the right speed. There we go. All right. And now I'm going to take away the text. All right, so let's bring ourselves the result of meiosis 1 through meiosis 2. I'm going to draw just one of these, right, for simplicity's sake, but know that both of these are going through the same processes. All right, so we're going to start with our original cell here. We're going to take this one through. I'm going to label it N2, here, meiosis 2. All right, so we're going to start with prophase. I'm not going to be able to draw the cell if I have it at that height. There we go. Prophase, and specifically prophase 2. What is happening in prophase 2? Remember, it's basically the same as mitosis. It's happening to the nuclear envelope. It is dissolving, yes. All right. Chromosomes are already condensed. What's happening to the mitotic spindle?
It begins to form.
Yeah, it's beginning to form again. All right, so let's draw that in. All right, so I'm taking this one through mitosis. All right, our next phase would be
Prometaphase.
prometaphase
2.
2, yes. So, prometaphase 2.
There's no late prophase?
We could draw like prophase in there, but it's not as needed because what doesn't happen here is crossing over.
Oh, I
Crossing
see.
over only occurs in prophase 1, does not happen again in prophase 2. Yeah, that's a good thing to be aware of, to know. I want you to know that. All right, so actually prometaphase 2, I don't have my nuclear envelope, it is completely dissolved. Right, my mitotic spindle is completely formed. Right, and my mitotic spindle is attached to each side of my chromosome. Sorry. So good, it's attaching in the same way that our mitotic spindle attaches to our chromosomes going through mitosis. Right? We don't have tetrads, we don't have to worry about tetrads, we're not going to draw in tetrads. Tetrads are already split. Alright, so after prometaphase 2, we have metaphase 2, yep. Alright, metaphase 2.
If you put your tongue at the roof of your mouth.
What is happening in metaphase 2?
If you put your tongue at the roof of your mouth, you could stifle a sneeze.
Yeah, they're lighting up. They're lighting up on the metaphase plate. Yes. So they're going to be lighting up, again, exactly the same way we've seen it in mitosis. Not much is different. All right. What comes next?
Anaphase 2,
Anaphase 2, yes, and what happens?
they split.
Yes, I'm liking these hand gestures. That's muscle memory, that's helping you remember what's happening here. Right, anaphase 2. Right, our centromeres are still, or our centrosomes are still at the poles.
Lego should make an interactive mitosis model.
They should.
Just because I think it'd be fun to repeatedly pull it apart again and again.
Yeah, in fact, when we do our exam review, I'm going to try to bring in some beads so you can physically do this out on your own. But again, our mitotic spindle, it's still attached, and it's the shortening of that mitotic spindle that has caused our sister chromatids to split now. Right? Because remember, our homologous pairs already split back in meiosis 1. So sister chromatids split. I'm actually going to write that up here because I think that is very important to know. And that's a huge distinction between meiosis 1 and meiosis 2. All right, after anaphase 2, we have
Telophase 2.
telophase 2, yes. All right, telophase 2, what is happening in telophase 2? What happens to our nuclear envelope?
It reforms.
It starts to reform. All right, our chromosomes have been completely pulled to the poles, and we can draw them within the nuclear envelope. They are starting to decondense. I will try to draw them a little decondensed here. Alright, so I'm trying to draw them decondensed so they're a little bit less coiled together, less rigid. Notice in our example that one of these nuclei are going to have chromosomes where crossing over affected them, and one nuclei does not. Right? That could be the case. Crossing over is kind of a random process too, helps create that genetic diversity. So what has to happen after telophase 2? Cytokinesis. All right, so then we're going to get 2 cells. Our nuclear envelopes are completely reformed. Diploidy level is haploid still. So I'm going to put a little N next to each of these. All right. Now remember, we had 2 cells that went through this process. We just looked at 1. So at the end of meiosis 2, we would have to draw in another 2 cells for that second cell that was the result of meiosis 1 that also went through this process. So I'm going to say, what happened here? I'm going to draw a little box around this one here. There we go. And for simplicity's sakes, I'm just going to try to draw it without crossing over so that you can get a visual that it is 4 cells that we have created here. There we go. All right. So end result of meiosis 2. We have 4 haploid daughter cells that have a different genetic makeup, right, than the mother cell, the parent cell, that originally started this process. And it's a different genetic makeup because of crossing over and because of independent assortment in metaphase. Right. Are there any questions? All right, Lucia.
Doesn't this continue to occur?
It could if we're still wanting to make more gametes. So what will happen to these cells, right? For these cells, this is the end of the line. They're not going to go through meiosis anymore. These cells are each gametes now, right? And they will be involved in sexual reproduction, in fertilization, right? So say we made egg and sperm here, right? An egg meets a sperm, we have a zygote, then we'll go through mitosis to create a new organism, a new individual.
That's right, I do find that fascinating, that zygotes are just potentials for any single organ. It's wild to think about.
Yeah. All right. So I do have a review slide up here. And I can pull this back up at the end of the lecture too if you need it. Right, this slide basically emphasizes everything that we've gone over. Right? Mitosis produces two diploid cells that are identical to the parent. Meiosis produces 4 haploid cells that are genetically unique. All right, I think I have a review slide next, potentially. I don't. All right. All right, so the whole purpose of meiosis, of sexual reproduction, is to create offspring that is has variety to it, right? It's genetically unique. So genetic material from your 2 parents are actually combined in this stage. Independent assortment along the metaphase plate is helping to increase that genetic variation, as well as recombination during prophase 1. Again, I kind of already emphasized this slide a little bit after my drawing there. All right, so that's why you might look similar to your parents, but you're not going to look identical to your parents. All right, now I should have a review slide for us to work through. All right, I want you to draw 2 different cells. All right, I want you to draw a cell in metaphase of meiosis 1 and metaphase of meiosis 2. And I want you to draw a cell, or answer some questions about a cell that has a ploidy level of 4n equals 8. Now, I haven't entirely explained this, so let me take this chance to explain what these numbers mean. 2n, we've seen that before. 2n means it's a diploid species, so 2 chromosomes per set, equals 6. 6 means there are 6 total chromosomes in this organism. So if I were to take 6 divided by its ploidy level of 2, I would get 3. Right? I know there are 3 chromosomes for this cell. Right? 3 chromosome sets, I should say. And each set contains 2 chromosomes, which equals 6. All right? I'm going to give you a chance to work through these questions. Let's see. We'll start with like 5 minutes and see where we are from there. And then, yeah, we'll go from there. I'm going to draw this out on the opposite side of the board, what 2n equals 6 would look like, to help give you a visual to get you started. Yes, in my exams, I typically do ask you to draw things out like this. This is great practice.
So, do the examples that you drew, would that be considered one pair?
It would be 2 pairs. There's 4 total chromosomes.
Oh yeah,
Two
because
pairs.
yes, because each X is
Yeah, so that would be 2n equals 4.
So are we supposed to list them?
They're supposed to be in metaphase 1 and metaphase 2. size indicating just one of the sets. Essentially, it's really kind of the same way that I've drawn it, except with another tetrad. But remember, if we're doing metaphase of Meiosis 1, what has occurred already in prophase 1? Okay, cycles have been formed. What is specific to prophase 1? It never happens in mitosis, it doesn't happen in crossing over has occurred. So you might be lined up on a metaphase plate, but you have to also indicate that crossing over has already occurred because it's already in metaphase.
So, good word I will put in for like set.
Yes, exactly. So my cell that I've drawn would be 2n equals 4 total chromosomes in 2 sets.
Hello, Meiosis.
2n equals 6 would be 3 sets, each having 2 chromosomes. So we still want sets of 2. Sets of 2. Yes. Well, we're getting there. I can see our mind is working in the right direction.
Yeah.
Right, so we have exactly what I've drawn, except for medium and middle. I've added another bivalent. Exactly. Yes, and if you were to draw it in color, you'd show color differences for crossing
Yeah.
over. If you were to draw it in pencil, you'd make a note that crossing over has occurred. Just some way to indicate that you know by metaphase crossing over. All right, so now take that cell, bring it through the test of meiosis 1 and prophase 2, and tell me what it would look like in metaphase of meiosis.
Yeah, you guys had to have four two sides of the
So these, would I have to make them yourself?
Not another cell, so it's like basically doubles. Well, tell me what do you mean by another cell?
Like I have 2 of these,
Mhm.
would I have to make another one?
You don't have to, you could just take one of the resulting 2 and bring it through meiosis 2. Remember, meiosis 1 creates 2 cells? Yeah. Actually, this is only just meiosis 2. So you could draw 2 cells going through phase 2, but they would be exactly the same. So you only really have to draw one. Pretty much. Now what's different about it?
That's 2 unequal to.
So this is it 2 anymore? It's no longer diploid where it happens inside, so it'd be n. And in this image, you're equal to.
Yeah, like most,
But if we're dividing our genetic material in half for 2 equals 6.
I think this would be cute.
6 divided by 2.
I really want these two.
So this is another stuff.
They're so cute.
Do I just have
But honestly, I
Yeah.
believe in that. Like, I don't know. I really like Boston. But like, then I'm gonna wear this on.
They have like almost.
Yeah. I'm sure they wear it. Like, I don't know. Like, I don't know, I don't like getting like, but it's like park, it's not like merch. Yeah, I guess that's true. But park is like,
We have 16 total chromosomes on it.
I just,
But
I'm just gonna do some shopping. Yeah, I've got the shoes. Yeah,
in my
me
car,
too.
and that's like
I
it.
have like 5 pairs of shoes.
Yes, like I'm not even
So
remember
what is
like
the
what
four?
shoes what am I doing? But I
how many chromosomes are in each set?
or something.
Yeah, I have literally.
Four, yeah, yep. And so we have 8 total chromosomes.
I want
Two sets of what?
like a crop trench.
Oh.
Two cells. It would basically be like instead of a bivalent, we'd have something like a tetravalent. Because each set we would have.
One and then like.
So we're never gonna have 16.
Yeah, that's.
But now so there's R has really good one.
Yes.
This one maybe did 7 after entrance.
Yeah, exactly.
I'm so stressed having a career because I need a pad.
Chromosomes are like,
I want to do all this stuff like on Christmas. I want to run courses for reproduction. I'm gonna need to share stuff on Is yours?
yes.
I think Lynn and I are flying out together and she's spending Christmas in Maine. Are you just celebrating Christmas a day early?
Yes, that's where it creates the most genetic diversity. But it's still independently in metaphase 2, right? Because maybe one side has had crossing over, one side hasn't. So it's still kind of random in the way it switches there.
But would that imply that in mitosis, they like not being according to each other?
They support it, but we don't get balance.
Yeah, I don't think
Because
either
when
of
those
you
guys
guys
know there's no,
Are you guys like together as a team?
there's not like a directionality per se to the chromosomes on the way that they would line up. Yeah.
Linnell told me that flying from out of Austin is closer. Yeah, it is. I'm flying from here and then from maybe sick over there. I went to my senior that's where that much of
You also
food poisoning all the way home. I don't know
yeah,
until
I
it's
heard
over.
this one. So they should be
That's brutal.
but
Like it's hereditary. So like when I was in the women's yeah, no, that's no, literally this is because you'll be coming from here to say Chicago. That's not you'll be flying over there and then straight. And also gross.
I was after like minutes.
Yeah, that would be serious. Or they wash like rare ones. So yeah, that would be cool. Yeah, yes, South Korea. Japan. Yeah, that's what I was gonna say. My friend went to.
I like that one.
Alright, I get the sense that we're all finished here. This is why I like to include timers on my slides, so it keeps me accountable a little bit. But I think we're all finished, so let's go over what it would look like. Alright, if we have a cell that is 2n equals 6, going through meiosis 1, how many bivalents would there be? There would be 3 bivalents, yes, because bivalent is a set of homologous chromosomes. Right? So if I were to line all these bivalents up in the metaphase plate, you should have drawn something similar to what I have up here. Right? Now we know we're in metaphase 1, so crossing over has occurred. Now I didn't illustrate that here, right? But you might have chunks that are slightly different colors on each chromosome to illustrate that crossing over occurred in the previous stage. All right. So that if I were to take this same cell, right, it's gone through the rest of meiosis 1, it's gone through prophase 2, and it's now at metaphase 2. How many chromosomes is there going to be in the cell? Or is there going to be in the cell? Are there going to still be 6? No, because in meiosis 1, we've gone from a diploid to a haploid cell. How many chromosomes will be left in these cells?
It would be 3.
It would be 3, yes. So after meiosis 2, and you likely only draw one cell, and that's quite fine, right? This one cell would divide into 2 cells, well, after the end of meiosis 1, and then each of those cells would go through prophase 2, and in metaphase 2, they would line up in the middle. Right? Notice the big difference here is the number of chromosomes. We've gone from diploid to haploid cells, and how they've lined up, right? We've gone from lining up bivalents to just lining up individual chromosomes. All right, so then I asked you a question about this cell that is 4n equals 8. What is the total number of chromosomes in this cell? See, fingers being held up, but it's 8, yes. So we have 8 total chromosomes. All right, how many sets of chromosomes do we have? We don't have homologous pairs in this case because we're not a diploid species. So how many sets of chromosomes do we have? We have 2, yes. All right, so you could have drawn this cell in a variety of different ways, right? So before DNA replication, right, we would still have 8 chromosomes. They're in 2 sets of 4 because they're a tetraploid species, okay? If you drew it after DNA replication, you could have drawn it like this cell on my far right, okay? Where it's in this X shape. They have sister chromatids to them, but we still see there's only 8 total chromosomes in 2 sets of 4 chromosomes each. All right, so I want to make sure that you understand how to interpret like 2n equals 6, or what does 2n equals 14 mean, right? Be able to interpret how many sets there are, right, and how many chromosomes are in each set. Now, if you know if they're a diploid species, we're talking about a homologous pair. If we're like, say, a tetraploid species, so 4n, we'd be talking about 4 chromosomes in a pair. Any questions? All right. I embedded our content check here, and I think that's going to lead us to the end of class today. So what I want you to draw is the steps of meiosis 1 for a diploid cell with 3 pairs of chromosomes. So working off of what we just did, and make sure to indicate where crossing over occurs. Now, if you want to use colored pencils, I actually remember to bring colored pencils with me today. You can grab them up front and make it over colorful if you'd like. That might help you remember crossing over.
All right, now I want to ask my question.
Oh, yes.
So I was thinking, so I didn't think about this during mitosis, but it came up to me during meiosis studies. But it's saying that now we have 2 nuclei. Now we have 2 nuclei. Can a cell, like, basically make any new organisms if, like, say, one of them magically disappears? Could it just create a new one from scratch?
Create a new one from scratch. What do you imagine?
I don't know. Let's say mitochondria disappears.
Okay. Okay.
It's like I get, like, would the cell be able to, like, replace it? Because if it can duplicate it, can it create one from scratch?
It should be able to, say if like an organelle were to disappear for whatever reason, as long as the genetic code that codes to create the mitochondria or the organelle is still present in the DNA, it could replace it.
Wouldn't it be this?
Yeah, that would be another big product. So now instead of just doing one stage, it's having you do all the stages. I'm not going to change this slide, but make sure you take a look at the homework slide. There is no homework, but I have linked a video that might help you preface our next lecture on transcription and translation. There's, again, a lot of terminology.
Punnett squares, and I feel like Punnett squares feel odd because I think about how much I've learned about my genetics, you know, because I'm close with many of my family members, and it becomes like, it almost becomes clear to me that the idea of is like, I don't think it's Punnett squares. I think we would look a little bit, I think they're a little bit more complicated.
They are. Punnett squares are very simplified versions of how genetics works.
I still think of codominance, and that one's always funny to me.
We'll learn about that.
Cats are the greatest example of it.
That is some of the parts of meiosis one.
Yeah.
It's missing a lot. Yeah.
But.
There would be 36 strings. So there's 3 pairs, but 6 total chromosomes.
Oh, so each one of these is just.
Exactly. Yep. So that cell we're looking at 2n equals 2.
Right here, this is one.
So 2 total chromosomes, only 1 homologous pair. It would start not with sets of 3, still with sets of 2.
This one, this one.
Yes. And then when we get to like prophase 1, you'll start lining them up into bivalence as they are in like homologous pairs at that point.
Yeah. So now I'm going to.
I like how you changed it, Grace. That looks good.
To this. Or
I would go through the individual slides. I know there's a lot of them. Well, they're condensed, so we're at least in prophase 1. But if we're in prophase 1, it show the nuclear envelope still disappearing. And also show that mitotic spindle is starting to form. So don't forget as you draw this, it's not just the chromosomes, it's also the nuclear envelope and the mitotic spindle that you want to indicate. Yeah. That looks like a good prophase 1. I don't even think we're gonna get that far. We'll have to look at chromosomal alterations next week. I guess that's so. One is squares. That won't be until next unit, but you still have to learn how we take DNA and make a protein. We're missing one phase though. Using the chalkboard for example. Go. First you're done the chalkboard drawing. Yeah. So we're not gonna have time to go through any more slides, so if you're done, you can make a pile up front. Here's where you go.
If I could think of
You don't want to read about it, but it's such an important stuff. Okay, that's just how you look at it.
But I got the same today.
Oh, these are the reasons. Yeah, that was. No, this is the document that. This is the one we got done. No, I was like talking to the office. I don't know.
First, this looks very, but then the anaphase, you were drawn with like both of them, isn't that when it pulls apart the chromatopsia?
It is, and maybe I didn't draw a big enough gap there. So what we
Like they
have,
separate the X is separated, right, from this one?
yes, the X's like would split in half there, and one X would go to each side. Yeah, even in chromatophase, I mean, this is like picky miss here. I would still draw them as like closer to each other because they're still bivalent. And then metaphase, bivalent line up, and then anaphase, yes, they would split.
So
Yep.
make them closer like this one almost?
Yeah, like I said, it's kind of a nitpicky thing, but it helps you remember like with the bivalent split. Who wants to go first? Chromatophage is like simple fibers are attached to the bivalents, but they're not lined up in a row yet. And then in metaphase, they're lined up in a row. Right, like they're attached to those chromosomes, but they haven't lined the chromosomes up on the metaphase yet. So again, instead of two bivalent splits, it would just be two. You would take what you've drawn from metaphase, if you would draw one down the middle and split it apart, that's exactly how you would draw it. Biggest thing is just like leaving a gap so I know that you are showing that it's split. Let's see. I'll change the metaphase, because the way you've drawn it looks like they split apart. And they're not split yet in metaphase, they're still bivalent, they're still together, just all in a row. And then the anaphase looks good. Yeah. So metaphase is the only one I would change. Just put them close together on a line in that middle. I'll be right back. Yes. I was gonna say, I didn't know I taught it spindle there, but you do late prophase or cassette. We're always gonna end up with in telephase one cell. Yes, number of chromosomes vary. Exactly, yes. When we get to the telophase part, you don't want 3 nuclei. You still just want 2 nuclei. Right, so we split it apart. Exactly. It's like if you were to draw a circle around these. Yes. And I didn't touch this. Lover's tugging last week. We just need to add in another chromosome pair of each of ours. So we have 3 pairs. So it's the 2 and 6. Looks good. I fixed my mother. I like it. Yep. Yep, that looks good.
On the other side ring. It's like if you go on each and then after everything, separate sides and around them, look at this rules and then yeah. And then right after that, it's why would move this?
So yours look like you had 3 on your end.
C-Y-T-O-K and
Did you originally put 2? Yeah, so I'm going to change to 3.
I've had practices over and over and over.
Yeah. No. I just did the data collection, like we did TS and stuff like that. Did you get out early? Like if you finished early. But do you have hers with me? Read a read. Did you guys have to read the Yeah, that took like a minute. And then the T-test and stuff, like if you know how to do it. So you have to do like a discussion.
And if you want to talk about it, we should talk about it tomorrow.
Did you have a camera?
You know how to sew?
It's so freaking long. You have to do like the wet lab, right?
Yeah.
Wet lab? Did you have a wet lab? No. Oh, I have a wet lab today.
Lucy, how's it going?
Tired.
Me too.
Thank you.
Thank you. I've done, I just, probably what we had. Something computer style and, I don't know, it's difficult to work with, so I almost feel like when I do it, I almost have to like brag about it too. It's like, I can't.
Yeah, I know. It's hard, especially if you don't have like a touch screen with a pen. Sometimes drawing, like meiosis on a computer is fitting.
I've done all kinds of drawings.
Yeah.
Ones, even free, both with freehand and with making shapes.
I'm gonna just
Yeah.
draw a bunch of these and like, I actually might take
Yeah,
a
I
photo
got
of these.
Yeah, I would say, you could ask ChatGPT to give you the ploidy level for a diploid cell. So it keeps changing the ploidy level. So maybe you draw a 2n equals 6, and then a 2n equals 8, and then a 2n equals 12.
I feel like we have to do a lot of studying. But our test is not next week, so next week, study.
Yeah,
Right?
yeah.
Our exam isn't.
So next week we still have more content, but there's no exam.
Yeah.
Yeah.
Alright, see you later.
Yeah, I'll see you later.
Our
Isn't our
exam is like next
tomorrow?
Thursday.
Or is it today?
It's today. Let me.
12:30, I think.
I'll look at my calendar quick. I have you at 12:30.
Alright, perfect.
Yes.
Oh, I burnt my hand from the tea.
Oh,
It's
ouch.
spilled. And oh, thank you. No, and
Yeah.
I'm walking, and everyone just like sees me spill the hot tea. 'Cause I'm like, ow. So, like that's embarrassing.
Eli, content is cooking?
Yes.
Good.
Not too difficult.
That's good.
Have a good rest of your day.
Yeah, you too.